Okay, the title of my presentation is Rational Design of Porphyrinoid Dyes for Photodynamic Therapy, Further Progress and Future Perspectives. My name is Professor John Mack. I am from Rhodes University, the Institute for Nanotechnology Innovation. So, photodynamic therapy, just to give a brief run through what that's all about. You have a photosensitizer dye. It is injected into the bloodstream. It accumulates in a cancer tumor. Then laser light is used to generate singlet oxygen, which destroys the cancer cells. And basically, if we're looking at this from the point of view of a Jablonski diagram, we photoexcite, there's intersystem crossing to the triplet state, and then reactive oxygen species can be generated either by energy transfer for singlet oxygen or electron transfer for radical species. The sorts of dyes that tended to be used for this are porphyrins, chlorins. Porphyrins, based because this comes from nature, so it's easier to gain approval in this context. But there's an issue with porphyrin dyes in that they absorb only weakly in the therapeutic window, which is from 620 to 850 nanometers. And what's behind the therapeutic window is that at shorter wavelengths, pigments within the body, like hemoglobin, can absorb, and at longer wavelengths, water starts to be an issue. So you have a window between about 620 and 850 nanometers where you have optimal transmission of laser light into the human tissue. Basically, background to this is that I have used molecular modeling to look at what happens to the frontier orbital energies of different porphyrin analogues when you modify the porphyrin ligand. And we're looking for ways that we can have a red-shifted and more intense absorption band in the therapeutic window. So the sort of dyes that we've looked at are chlorins, chirols, and N-confused porphyrins. And our starting point was to look at a tin-4 porphyrin with thiophene as at the meso-arrow positions, and the thought being that having the transaxial ligation would help to limit aggregation, which can be a problem with photosensitizer dyes. And we got favorable results in this context. So after that, we started moving forward towards chirols. And in a similar way, we obtained favorable results, but with a more red-shifted and intense Q band in this context, I should say. We've also looked at bromination of porphyrins as a way to redshift the spectral band and enhance its photosensitizer properties. We've looked at chlorins, where you've got a reduced peripheral pyrrole bond in a similar manner to chlorophyll, and we obtained a redshift to around 660 nanometers, a more intense absorption band, and favorable results not just for PDT, but also for antibacterial studies that's referred to as PACT. And again, we're looking at the tin-4 complex and obtained highly favorable looking PDT activity results in that context. And the last type of porphyrin analog that we've looked at in a big way is N-confused porphyrins, where one of the porphyrin rings is flipped to the outside in terms of the pyrrole nitrogen, and that shifts one of the absorption bands out into the 700 to 800 nanometer regions, deep into the PDT window. We used molecular modeling to explore the reasons for that, and obtained promising looking PDT activity results in that context. And we've looked at what happens if we extend the pi system of N-confused porphyrins, and basically we can shift the band out almost to 900 nanometers in that context and obtain, we've looked at molecular modeling again to understand the trends and obtained reasonably positive looking activity results in that context. In the last year, we've continued to look at corals with the help of researchers in China. One of the students, Rhoda Soy, formed a A2B type coral with a quaternized nitrogen atom, and that helped to enhance both the PDT and the packed activity properties. We've also continued to look at chlorines as well, and the big focus of what we've been looking at recently has been to add functionality which helps to target mitochondria, because even under hypoxic conditions, which tends to be the case with cancer cells, mitochondria retain a high oxygen concentration, and that helps to generate singlet oxygen. And we obtained favorable results when we added a mitochondria targeting moiety to porphyrin, and we're going to extend that to other porphyrin analogues in future. Where does CHPC help? Really with the large memory allocations that are provided by CHPC, but by the time you have a molecule as large as the one shown on this slide, it really is important to be able to have 45 gigabytes is what we tend to use by that point to be able to run the TD-DFT calculation properly. So CHPC definitely really helps in this context. Another area where we've started pushing what we do with CHPC is we've been looking at how we can use molecular electrostatic potential maps to rationalize trends that we observe in experimental properties. Here we were looking at a series of coorbidipides and the packed activity properties, and we found that looking at the dipole moments and the electrostatic potential maps really helped in that context, and CHPC again helped to push things along in that context. So you can read the conclusions on the slides that I have also uploaded. And acknowledgements, I have to thank Prof. Bello Nierkong and Rhodes University for the opportunity to carry out this research at the Institute for Nanotechnology and Innovation, and for funding, Rhodes University, African Laser Centre, NRF, and also we get support for the packed research that I discussed from MinTech. And with that, I would encourage you to check my slides, which have also been uploaded.